Hello, so let's continue on with cellular respiration. So you should have at this point watched the first part of the lecture video. You should have watched the Amoeba Sisters video about cellular respiration, and you should have the chart filled in at least partially, if not all the way, about what happens in glycolysis, the preparatory stage, the Krebs cycle, and oxidative phosphorylation. And we're going to go into it in a little bit more detail, so as we're going through it, if there's anything that you think you might have missing in your chart, fill that in. Most of the questions for this quiz and exam for this chapter are going to come off of that chart. So really I just want you to be able to complete that chart from memory, and that is enough for me for this chapter. Okay, so we have these three stages of the cellular respiration. And we had the Krebs cycle and we had glycolysis that made a little bit of energy. But most of the energy is coming during oxidative phosphorylation. Oxidative phosphorylation refers to phosphorylating ADP into ATP during the final stages of the electron transport chain. So that is oxidative phosphorylation. If we're talking about anything else, it is not oxidative phosphorylation. Oxidative phosphorylation is what happens as a result of chemiosmosis following the electron transport chain. During glycolysis and the citric acid cycle, that small amount of ATP we made was made by substrate level phosphorylation. So that means we had some substrate that we pulled a phosphate off of, put it onto ADP, and made ATP. That's substrate level phosphorylation. That's how we made the ATP in glycolysis and the citric acid cycle. So let's go through these steps. Step 1, glycolysis. You start with glucose. At first, you have to use some energy. It's going to take a little bit of ATP in the first stage. Then in the second stage, we get a bunch of ATP. We also generate some NADH. And remember what NADH is doing? It's carrying electrons. And we create that ATP and the NADH as we are breaking glucose in half, making 2-pyruvate. So if you remember, in glycolysis, glucose is broken down into 2-pyruvate. 4 ATP are formed, 2 ATP are used, so overall we have made 2 ATP. That is a little bit of energy that your cell can right now go and use. We also took 2 NADH we took 2 NAD plus and made 2 NADH. Are those oxidized or reduced? Stop and think for a second. They picked up electrons. That means that they are reduced. So NAD plus is reduced to NADH. Those electrons are carrying a heck of a lot of energy. So they're carrying a lot of energy as NADH. It's just holding on to it right now. It's not yet able to use that energy in our cells. It's holding on to that energy until it gets to the electron transport chain where it feels safe enough to create ATP. Glycolysis will occur whether oxygen is present or not. So if you have a cell that suddenly loses oxygen, it can still continue doing glycolysis through a process called fermentation that we'll discuss later. If oxygen is there, it's not going to go through fermentation. It's going to keep going through, it's going to continue being oxidized. So in the presence of oxygen, pyruvate is going to enter a mitochondria if we're talking about a eukaryotic cell. If we're in prokaryotes, this is still in the cytosol. And glucose is going to be oxidized completely in the mitochondria. The first stage is called the preparatory stage. Pyruvate is going to be converted to acetyl-CoA. So we have pyruvate with its 3 carbons. It is going to be decarboxylated. So this carbon is removed. It's just going to float off as carbon dioxide. That leaves acetate. It's also going to take a couple of electrons and reduce NAD to NADH. So NADH is holding on to a lot of electrons until it feels safe enough in the electron transport chain. Coenzyme A is going to attach to this acetate. So this acetate, this coenzyme are going to attach to make acetyl-CoA, and acetyl-CoA is going to go into the Krebs cycle. So the citric acid cycle or the Krebs cycle is going to take this acetyl-CoA and is going to eventually take these 2 carbons and break them off as 2 carbon dioxide. So at this point, glucose is completely oxidized. We have completely taken all those electrons off of it. We've broken those bonds. Carbon dioxide is oxidized. It is oxidized by making NADH, NADH, and FADH. If you remember, those are carrying electrons, so they are holding on to those high energy. NADH and FADH2 has a lot of energy in those electrons, but it can't use it yet. It has to get to the electron transport chain before it can turn it into energy. So, for every pyruvate, we have made 1 ATP right here. We made 3 NADH here and here. We made 1 FADH2. But if you remember, glycolysis broke into 2 pieces. We have 2 acetyl-CoAs. So we're running this cycle twice. Okay? So for every glucose, we're going to make 2 ATP. We're going to make 6 NADH. We're going to make 2 FADH2 and 4 carbon dioxides that you just breathe on out. So we've now got a lot of NADH and FADH2 that are holding so much energy, and they just can't do anything with it yet. So citric acid has 8 steps, and I'm not going to make you memorize any of that. Know this, okay? If you can tell me how many ATP, NADH, FADH2, and carbon dioxide you make, you can be able to tell me for each acetyl-CoA, and you can tell me for each glucose. So tell me for glucose, you just double these numbers, right? You're running it twice. Let's skip that. So we have a lot of NADH and a lot of FADH2 that so desperately want to let these electrons turn into energy. So these electron carriers are going to take their electrons to the electron transport chain. And the electron transport chain, we're going to pass those electrons through carriers. We're transporting electrons, and that allows the energy in the electrons to slowly be released. Then that slow release of energy is going to allow ATP to be made by oxidative phosphorylation. So this oxidative phosphorylation has 2 parts. One is the electron transport chain, when we're passing electrons and taking their energy, and one is chemiosmosis, when we're making ATP. So in the electron transport chain, this is happening in the cristae of mitochondria, or in the plasma membrane if you're in a prokaryotic cell. This has to be in a membrane because all these proteins are in a membrane. And it's going through a whole bunch of complexes. We're not going to go through what all these complexes are. But they're going to, NADH is going to pass off its electrons. So this is going to be reduced. It's going to pass it off, so it's oxidized. Then this is reduced and then oxidizes. It's passed it off. Reduced and then oxidized, okay? So everything's being reduced and oxidized. Reduces and oxidizes. It's passing down these electrons. And it's going, as the electrons are moving down, they're slowly releasing energy. Eventually those electrons have lost all of their energy. They're released from the electron transport chain, picked up by oxygen where they turn into water. As these electrons are passing down and it's slowly releasing energy, the energy that is released is not just going to dissipate away. We are going to capture it. And the way we're going to capture it is by taking hydrogen and pumping it out of the cell. So as this picks up electrons, it passes down the electrons and then uses that energy to pump out hydrogen ions. So hydrogen ions means we're going to have a lot of hydrogen end up outside of the cell and not as much left inside. So we now have a gradient. And cells don't like gradients, do they? Cells like equilibrium. So all this hydrogen that moved outside of the cell is going to come back down its concentration gradient. It's going to come through this molecule called ATP synthase. ATP synthase, that is an enzyme you need to know, is going to spin around. And as it spins around, every time hydrogen comes through, it spins around and takes ADP and inorganic phosphate and combines them into ATP. And you have just made a ton of ATP. It's worth noticing that NADH drops off its electrons here, and FADH drops off its electrons here. So NADH, those electrons are going through more enzymes, so they can pass out more hydrogens. FADH is going through fewer enzymes, so they can pass out fewer hydrogens. So for every NADH that drops off its electrons, we will make 3 ATP. For every FADH2 that drops off its electrons, we make 2 ATP. That is something you need to memorize. Okay, so this is a proton motive force. This is how hydrogens can do work. So as you have electrons, You pass down the electrons, pump out hydrogen. Pass down electrons, pump out hydrogen. Pass down electrons, pump out hydrogen. So for every NADH, you've had 3 opportunities to pass out hydrogen, so you can make 3 ATP. For every FADH2 starting here, you have 2 opportunities to pass out hydrogen, so you have 2 ATP that you can make. That proton motive force means that because you get a bunch of hydrogens out here, it's going to move, and when it moves down its concentration gradient, it causes ATP synthase to move and creates ATP. So our energy has flowed from glucose to NADH, or FADH2, to the electron transport chain, which creates a proton motive force and generates ATP. Again, 3 ATPs for every NADH and 2 ATPs for every FADH2. It went through glycolysis, then pyruvate oxidation, then the citric acid cycle, then oxidative phosphorylation, which is both the electron transport chain and chemiosmosis. Okay, most of that cellular respiration depended on oxygen to pull electrons down the transport chain. Oxygen is really electronegative, so those electrons are willing to move through that transport chain. Without oxygen, the electron transport chain would stop working. So all of this would just back up. It would fill up, electrons would stop working, it would be done. You'd be stuck with your NADH. So without oxygen, you're going to stop oxidative phosphorylation happening. You're eventually going to back up and stop the citric acid cycle from happening. But glycolysis could keep happening if you could get more NAD. So all of your NAD is turned into NADH waiting for oxidative phosphorylation, your cell's going to have no way to get energy. So instead, glycolysis is going to go to anaerobic respiration. Anaerobic respiration means that you can do all of this without oxygen. It's going to find some other way to pull things through the electron transport chain. Or it's going to go through fermentation. Fermentation is going to use substrate level phosphorylation instead of an electron transport chain to make ATP. In fermentation, you are going to keep glycolysis running, but then you're going to have extra steps that turn your NADH back to NAD+. So all that NAD that you just made is not going to be used for energy. It's just going to be recycled back to NAD+, so you keep glycolysis running. So it's not as energetically favorable. You're only going to make a couple of ATPs from glucose, and that's it. Through fermentation, there are 2 types of fermentation. One is alcohol fermentation that makes ethanol and carbon dioxide, and lactic acid fermentation, which makes lactate or lactic acid. Alcohol fermentation is used by a lot of yeast. So if you want to make beer or wine, because sometimes adults want to make those things, that you just make your yeast grow without oxygen, and they're going to go through alcohol fermentation. Animal muscle cells can go through lactic acid fermentation, which is why sometimes if you've been exercising a lot, your cells are needing to make a lot of energy, but you're not, you're breathing really fast, but you just can't quite keep up with it. Your muscles might get a little sore from that lactate buildup. And then lactate can be converted back to pyruvate. Okay, let's watch a little video.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are 2 types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
Okay, and then again, this is only getting those 2 ATP from glycolysis, so your cells are not going to grow very well in this process. Your cells, as soon as oxygen is available, they're going to switch back to cellular respiration. Fermentation is just a don't die until oxygen is available. It's just to keep yourself making a little bit of ATP to survive until that oxygen is available again. Okay, so all of these processes, anaerobic, aerobic, and fermentation, all use glycolysis. And they all oxidize glucose, and they all get energy from food. In all three, NAD+ is an oxidizing agent that picks up electrons during glycolysis, although they have different processes for oxidizing NADH back to NAD+. So in fermentation, either pyruvate or acetaldehyde, some organic molecule is going to pick up and act as a final electron acceptor. And that's the end of getting energy from those electrons. In cellular respiration, electrons are transferred to an electron transport chain. If it's aerobic respiration, the electron transport chain ends with oxygen picking up the electrons. And if it's anaerobic respiration, the chain ends with something that's not oxygen picking up those electrons. In cellular respiration, you're going to have about 32 ATP per glucose, and in fermentation, you're going to have about 2 ATP per glucose. So let's talk about some different types of cells. We are obligate aerobes. We only grow in the presence of oxygen. That's the only way we can live. Without oxygen, we don't live for very long. But there are organisms called obligate anaerobes. Obligate anaerobes will either go through fermentation or anaerobic respiration. They only live without oxygen. If oxygen is available, oxygen actually kills them. There's also facultative anaerobes. Facultative anaerobes grow when oxygen is present. They prefer oxygen and grow really well. But if oxygen is not available, they can either go through fermentation or anaerobic cellular respiration to survive. So in a facultative anaerobe, pyruvate is kind of this fork in the road, that if oxygen is present, it goes through aerobic cellular respiration. If oxygen isn't present, it'll either go through fermentation or anaerobic cellular respiration. Glycolysis is very ancient. We know that because it's used in almost all cells. Early prokaryotes were probably using glycolysis before there was oxygen in the atmosphere. It's used in both cellular respiration and fermentation. It's the most widespread metabolic pathway on Earth. So this is happening in more cells than any other metabolic pathway that there is. It occurs in the cytosol, so it doesn't require any membranes. And then glycolysis and the citric acid cycle are major intersections to various catabolic and anabolic pathways. So that means that glycolysis and the citric acid cycle can be used actually to build things through anabolism by making them run backwards from how they run in catabolism. Let's see. So a lot of words. Let's see what we really care about. So catabolic pathways take electrons from organic molecules. So we talked about specifically what happens to sugar, but it can also take electrons from proteins and fats and use them in cellular respiration. If protein is used for fuel, it's going to be broken into amino acids, and then amino acids can go into, all these different steps. Fats can be digested to glycerol and fatty acids, which can also be inserted into the process. Fats actually hold a lot of energy in their bonds, and 1 gram of fat is worth almost twice as much energy, twice as much ATP as carbohydrates or proteins. We can also use small molecules from food to build things. This is called biosynthesis or anabolism. These small molecules can come directly from food, from glycolysis, or from the citric acid cycle. So a metabolite is something that is necessary for metabolism to happen. And cofactors can be non-proteins that are needed for enzymes to function, either inorganic cofactors like ions or metals, and organic cofactors like vitamins. So that just means that these, instead of running forwards and breaking it down, we're actually going to move backwards and build up things that our body needs. So build DNA, build sugars, build the stuff. So you don't have to always just break your sugar all the way down. And then feedback inhibition, we've talked about this a couple of times, so I'm going to go through this pretty quickly. Feedback inhibition is the most common mechanism to control your metabolites. If ATP concentrations start to drop, respiration speeds up. It means you need ATP, so you're going to start doing respiration. But if there's plenty of ATP, respiration slows down, and you start using that ATP for anabolism. So control of catabolism is based on regulating the activity of enzymes at strategic points. So we control whether it's moving forwards through catabolism or stopping and allowing anabolism by regulating these enzymes. If you stop the enzyme, it stops the whole path, so you're not making any more ATP. Okay, that was a lot of information. I hope you're really coming to class for these chapters because we're doing some activities that make it a little bit easier to know what we're talking about. But please make sure that this form, you can complete completely. Okay? At this point, you should be able to tell me about glycolysis, the preparatory stage, Krebs cycle or citric acid cycle, and the electron transport chain, and oxidative phosphorylation, and about fermentation. You're going to tell me where it occurs in a prokaryotic cell, where it occurs in a eukaryotic cell, how much ATP is made, how many NADH or FADH2 are made, and other important details that we talked about in class. Okay, I will see you later. Bye.